Usually, when you think about getting a medical diagnosis, there's this expectation of precision, right? You break a bone, they take an X-ray, and boom, there's a jagged white line. The attending just points right at it.
Right, yeah. It's binary, it's actionable, broken or not broken.
Exactly. And it gives you this very comforting sense of certainty.
Well, I mean, in structural medicine, seeing the problem is really like 90% of the battle.
But the moment you step into endocrinology or, autoimmune diseases, that certainty just completely vanishes. The landscape becomes incredibly murky.
Oh, absolutely.
Like with type 1 diabetes, where your own immune system is systematically destroying the insulin-producing beta cells in the pancreas.
Yeah.
There's no simple X-ray for that.
No, not at all. You're dealing with this microscopic cellular destruction that's hidden deep inside a major organ.
Right. Which means you're flying blind. And that brings us to our mission for this deep dive. Whether you are, say, a medical student grinding through your endocrinology block, a clinical researcher, or just someone fascinated by bioengineering, we are breaking down the absolute cutting edge from the book Type 1 Diabetes, Methods and Protocols.
It's a fantastic resource, honestly.
It really is. Our goal today is to give you a comprehensive, scientific, but accessible breakdown of how we visualize, engineer, transplant, and ultimately protect these beta cells. So, okay, let's unpack this.
Let's do it. And I think to really grasp this, we have to start with the anatomical nightmare that is the pancreas itself.
Right, because the beta cells aren't just sitting in one neat little lump.
Exactly. They live inside the islets of Langerhans, which are these tiny clusters of cells. But those clusters are scattered by the thousands all throughout the exocrine tissue, the part of the pancreas that makes digestive enzymes.
So historically, if researchers wanted to know how much beta cell mass a patient or a lab mouse had left, the traditional method always seemed so flawed to me.
Oh, the stereological assessments.
Right. They would just take a few random 2D microscopic slices of the pancreas and then basically use math to guess the total volume based on what was in those slices.
The sampling bias there is just staggering. Because the islets are so heterogeneously clustered, taking a 2D slice is kind of like, well, it's like taking a single core sample of a huge mountain range and assuming the entire state has that exact same mineral distribution.
You could drill into a dense pocket of islets or just hit a complete dead zone.
Exactly.
Yeah.
So your resulting data on beta cell mass was often just wildly inaccurate. But the book details this incredible shift to 3D optical molecular imaging to fix this. Specifically, they use light sheet fluorescence microscopy, or LSFM.
And the physics of this is wild. They use a technique called tissue clearing, right? Because normally a pancreas is totally opaque.
Right. It's opaque because the water and the lipids inside the cells have different refractive indices. So when light hits the tissue, it scatters everywhere. You can't see through it.
So how do they clear it? Do they just dissolve the fat?
Essentially, yes. The protocols systematically strip the lipids right out of the exocrine tissue, and they replace the water with a solvent that perfectly matches the refractive index of the remaining proteins.
So the pancreas literally becomes transparent.
Like a piece of glass. It's amazing.
Yeah.
Then you introduce fluorescent antibodies that only bind to beta cells.
Wow.
Yeah. And when you put that under the light sheet microscope, you can illuminate and map the exact 3D location of every single islet in the entire organ, down to micrometer resolution.
So it completely eliminates the mathematical guesswork. You're just seeing the actual total mass.
Precisely. But, tissue clearing is an ex vivo process. You can only do it in the lab on a removed organ. Visualizing beta cells inside a living, breathing patient is a totally different ballgame.
Right, which brings us to in vivo PET imaging. And this part blew my mind. They are literally repurposing neuroimaging tracers to look at the pancreas.
Yeah.
It's like finding out the pancreas is moonlighting as a brain. It's just borrowing neural receptors to do its job.
That is a great way to put it. And the biological reason for that crossover really comes down to evolutionary efficiency. Beta cells are neuroendocrine cells.
Meaning they act like neurons.
In many ways, yes. They share a lot of the same cellular machinery. A beta cell synthesizes insulin, packs it into little secretory vesicles, and releases it when triggered. Mechanically, that is exactly how a neuron packages and releases neurotransmitters, like dopamine.
So because they use the same physical machinery, we can use the same radioactive tracers in a P scan.
Exactly. The book highlights a tracer called 18FFP plus DTBZ.
Very catchy name.
Right. Rolls right off the tongue. But that molecule was originally designed to find VMAT2, which is the vesicular monoimmune transporter 2. Neurons use VMAT2 to pump transmitters into vesicles.
The beta cells use it too for insulin.
Yes. So injecting this tracer lights up the beta cell mass on a PET scan. We see the exact same thing with another tracer, 11C plus PHNO, which binds to dopamine D2 and D3 receptors.
Which, again, beta cells happen to have.
Exactly. So for clinical applications, if you scan a type 1 diabetes patient over time, you actually see a reduced uptake of these neural tracers in their pancreas. It gives clinicians a quantifiable, direct measurement of their beta cell loss. And they're also doing this with GLP-1 receptor PT imaging to evaluate mass and even localize insulomas.
That is so cool. But mass is only half the story, right? Just because the cells are there doesn't mean they're actually working.
Ah, exactly. Function is critical.
And the book outlines this really clever way to measure function using MRI and zinc, because insulin isn't just floating around inside those vesicles. It's stored in a crystalline form, literally bound to zinc ions.
Right, which means when the beta cell secretes insulin, it has to secrete the zinc along with it. They're inextricably linked.
So when your blood sugar spikes, the beta cell fires, and it dumps this surge of free zinc into the extracellular space.
And researchers have designed gadolinium-based MRI contrast agents that only react to that extracellular zinc.
So the MRI scan stays relatively dark until the moment the beta cell actually fires.
Exactly. Once that zinc hits the contrast agent, it alters the magnetic relaxation properties, and you get this bright functional hotspot on the MRI in real time. You're literally watching the physiology happen.
It perfectly bridges anatomy and physiology. But, okay, let's say we've mapped the deficit. We know the cells are gone. The clinical reality now is we have to replace them.
And that introduces some massive biological and engineering hurdles.
I'll say, just extracting human islets from a donor pancreas sounds like a nightmare. It requires these super strict GMP, good manufacturing practice facilities.
It is an incredibly harsh process. First, you have to perfuse the donor organ with a cold preservation solution, like UW solution, to stop ischemic damage.
Right.
You have less than 8 hours of cold ischemia time.
And then you basically have to digest the organ
Rah.
using collagenase enzymes.
Right. And that enzymatic digestion is the biggest vulnerability. The pancreas is glued together by a dense extracellular matrix. You use enzymes to melt that glue, but if you leave them on even a little too long, you overdigest the tissue.
Stripping the islets of the surface receptors they need to survive.
Exactly. They won't be able to engraft in the new patient.
It's like trying to safely extract delicate berries from a thorny bush while blindfolded.
That's a very accurate analogy.
And even if you pull it off perfectly, you still have this huge geometric problem. Because natural islets vary wildly in size from like 50 to 400 micrometers.
Which is a huge issue for oxygen supply. Inside the donor pancreas, a massive 400 micrometer islet does fine because it has its own internal capillary network delivering oxygen directly to the center.
But when you isolate it, you rip all those blood vessels off.
Right, so a transplanted islet has to rely completely on passive oxygen diffusion from the surrounding tissue until new blood vessels can grow in a process called angiogenesis, which takes about 10 to 14 days.
And oxygen can only physically diffuse through tissue for about 150 to 200 micrometers.
Exactly. So if your islet is 400 micrometers wide, the oxygen just can't reach the core.
So the center of the islet literally starves to death.
Yes. It undergoes severe hypoxia and central necrosis long before the new blood vessels arrive. We lose a massive percentage of the donor graft in the first 2 weeks just because of this diffusion limit.
So how do we fix nature's geometry? The text talks about engineering our own uniform structures, right? Pseudo-islets.
Yes, size-controlled 3D multicellular organoids. The methodology is brilliant. They take those very donor cells, break them all down into single cells, and force them to reaggregate in special culture plates.
And they intentionally design them to be perfectly uniform, staying strictly under that 150 micrometer limit.
Exactly. Every single cell is guaranteed access to oxygen. And they don't just use beta cells either.
Right, they mix in human amniotic epithelial cells, HAECs.
Which is crucial. HAECs have incredible anti-inflammatory and immunomodulatory properties. By co-culturing them directly into the pseudo-islet, you're building a graft that secretes its own local defense cytokines to suppress the host's T cells.
Okay, so we've engineered these perfect, uniform, self-defending pseudo-islets. Do we just drop them into the patient's bloodstream and hope for the best?
Definitely not. Where you put them is just as critical. Historically, clinical transplants are infused into the liver via the portal vein, but it's an awful environment.
The blood flow is super turbulent, right? And
Yeah.
the oxygen is low.
Yes, and the immune response there is immediate and severe. So the book explores some really creative alternative sites. For small animal models like rodents or monkeys, they actually transplant the islets into the anterior chamber of the eye.
Wait, really? Into the eye? That sounds radical.
It does, but it exploits something called anterior chamber associated immune deviation. The eye is an immune privileged site. The body doesn't mount its usual aggressive inflammatory response there.
Oh, so the graft can survive without dumping heavy immunosuppressants into the patient.
Right. Plus, the cornea is transparent. It acts as a built-in natural window.
Oh, wow.
Yeah. Researchers can use microscopes to just look right in and non-invasively track the survival and function of a single islet over months.
That is an amazing in vivo laboratory, but obviously you aren't doing eye transplants for large-scale human therapies.
No. No. For large animals and eventually humans, the book details this elegant surgical approach, the composite islet kidney transplant model.
And this solves the IBMR problem, right? The instant blood-mediated inflammatory reaction.
Yes. IBMIR is devastating. If you put naked islets straight into the blood, the tissue factor on the islet's surface instantly triggers the host's blood to clot around it.
It just traps the graft in this huge inflammatory matrix and destroys it in hours.
Exactly. So the composite kidney model uses autologous prevascularization to bypass IBMIR entirely. Surgeons actually transplant the islets under the renal capsule of the donor's kidney weeks ahead of time.
So they let the islets completely heal and grow a mature capillary network that connects to the kidney's own blood vessel.
Right. The islets are no longer free-floating naked tissue. They are a fully integrated part of a solid organ.
And then they take that whole kidney acting like a Trojan horse carrying the hidden islets and transplant it into the recipient.
Yes. The surgeon just hooks up the major renal artery and vein. The islets get immediate, rich oxygenation without ever directly touching raw, turbulent host blood. IBMIR is never triggered.
I love that Trojan horse analogy. It totally bypasses the delayed revascularization issue. But wait, what about tracking grafts non-invasively if they aren't in the eye? Like naked or encapsulated islets using standard clinical scanners.
Right, the text does touch on that. For microencapsulated islets, they're developing techniques using clinical CT, ultrasound, and 19F MRI to monitor them non-invasively. But for research models, the gold standard for tracking viability is in vivo bioluminescence imaging, or BLI.
Using firefly enzymes, right? Luciferase.
Exactly. The stem cell-derived islets are genetically modified to express luciferase. You inject the substrate, luciferin, and the cells emit light. But the beauty is that the chemical reaction requires ATP and oxygen.
Oh, so it's a direct proxy for cellular metabolism. If a cell is dying from hypoxia, it makes no ATP, so it emits no light.
Precisely. You just use sensitive cameras to quantify the light, and you get a perfect survival trajectory of the living graft.
Okay, here's where it gets really interesting. We've solved the macrobiology, the geometry, the surgery, but the protocols in this book push it down to the molecular level using nanomedicine.
Yes, because you still need to protect the graft from ongoing autoimmune attacks and encourage the beta cells to multiply. This is where microRNAs come into play.
RNAs, right.
Right. They are these tiny, non-coding RNA molecules, just 19 to 22 nucleotides long, but they act as master regulators. They bind to messenger RNA and either destroy it or stop the ribosome from translating it.
They basically control everything in the beta cell, from ATP generation to apoptosis or cell death.
Yes, they're incredibly stable in serum, which makes them great biomarkers, but the book focuses on using them as therapies through theranostics, a fusion of therapy and diagnostics.
I love theranostics. It's like having a GPS tracker that also repairs your car's engine while you're driving.
That is exactly what it's like. They use superparamagnetic iron oxide nanoparticles, SPNs. They chemically attach custom oligonucleotides to these iron particles designed to target specific miRNAs.
So on the therapy side, the beta cell swallows this particle, and the oligonucleotide alters its genetics to promote proliferation and shut down cell death.
Right. You're actively instructing the graft to heal itself. And on the diagnostic side, that iron oxide core creates a massive magnetic disturbance.
So when you put the patient in an MRI, the particles show up as these dark, hypointense spots. You can physically watch the drug being delivered to the pancreas.
Exactly, and the engineering of the delivery vehicles is just as wild. They use micelle-based nanocarriers made of amphiphilic block copolymers.
Okay, amphiphilic meaning one end loves water and the other end hates it.
Yes, a lipophilic core and a hydrophilic shell. When you put them in the bloodstream, physics forces them to self-assemble. The water-hating tails collapse inward, creating a safe core for the delicate RNA drugs.
And the water-loving heads form a stealth shell on the outside to hide from the immune system.
Exactly. It's targeted nanoscale delivery. They even use magnetic particle imaging MPI along with machine learning to track these iron-labeled islets post-transplant, which gives a much clearer signal than MRI because it only detects the iron with no background tissue noise.
Wow. They also mentioned using dextrin-coated iron oxide particles with SIRNA to inhibit apoptotic genes, right?
Yes, another great example of silencing the pathways that lead to graft death.
But, to prove any of these cellular or nanoscale therapies actually work systemically, you have to measure the patient's whole body insulin sensitivity.
Right, and that requires the gold standard measurement, the hyperinsulinic euglycemic clamp, or HEC.
The HEC. The logic behind this clamp is just brilliant to me because normally your blood sugar is this chaotic mess of your liver pumping out glucose and your muscles absorbing it.
Exactly, it's too noisy. To measure true peripheral insulin sensitivity, you have to completely shut down the liver's glucose production.
So how do you do that? Just flood the system with insulin?
Yes, you infuse a continuous, massive, supraphysiological dose of insulin. The liver instantly stops making its own glucose.
But if you just did that, the rat or patient would die of severe hypoglycemia.
Right. So simultaneously, you run a variable glucose infusion, you constantly monitor their blood, and adjust the glucose drip to keep their blood sugar perfectly normal, or euglycemic.
So by flooding the system with insulin, you force the body to stop making its own sugar,
Yep.
and then you see exactly how much external sugar it takes to keep the engine from stalling.
Exactly. Because the liver is offline, the exact rate of glucose you're infusing mathematically equals the exact rate the peripheral tissues are consuming it. It's a flawless metric of insulin sensitivity.
And the book stresses doing this on conscious rats, right?
Yes, using the tail artery for sampling and the tail vein for infusion under local anesthesia. General anesthesia totally messes up metabolic rates, so keeping them awake is crucial for accurate data.
And they apply this rigor to tracking complications too, like diabetic retinopathy.
Oh, yes. Retinopathy is linked to lipid abnormalities, but a mouse retina is so incredibly tiny. Traditional mass spectrometry needs chemical derivatization, adding tags to the lipids, but you lose too much sample doing that on something so small.
So they use an LCMS, liquid chromatography mass spectrometry method that doesn't need derivatization at all.
Right. It's highly sensitive and lets them accurately measure cholesterol and oxysterols in these microscopic retinal tissues.
It's just staggering. When you look at the whole picture, we're moving from 2D stereology to 3D LSFM clearing. We're engineering pseudo-islets with HAECs instead of just isolating varied natural islets.
We're bypassing IBMR with Trojan horse kidney grafts, manipulating genetics with miRNA theranostics, and proving it all with the HEC clamp.
It's an absolute masterclass in bioengineering.
It really is. And for the medical students and learners listening, I want to leave you with a quick review exercise.
Oh, a pop quiz. Love it.
Just a thought experiment. Based on our discussion of theranostics, if you were designing a nanoparticle to protect a newly transplanted islet graft, what two distinct capabilities must your nanoparticle possess? And what specific imaging modality would you use to track iron oxide-based particles in vivo?
Feel free to pause and test your recall on that one.
And beyond just diabetes, consider the broader implications. If we can successfully camouflage beta cells inside a kidney and manipulate their genetics using traceable nanobots, the future of type 1 diabetes treatment might not look like diabetes management at all.
Right. It might look like targeted organ augmentation.
Exactly. Consider what other localized autoimmune diseases like rheumatoid arthritis or multiple sclerosis could be cured using this exact same blueprint.
A completely new paradigm for medicine. Well, thank you all for joining us on this deep dive. Good luck with your medical studies and clinical practice, and we'll catch you on the next one.